And
All right, let's get started. So the first speaker of this session is Kenan.
thank you. Thanks a lot to the organizers. I'm really excited to be here for the breadth of what I've seen so far. It's been really helpful to my thinking. And as you'll see, I've titled my work sort of towards physical learning, because I think what I'll hopefully discuss with you today is I'm interested in setting the stage for understanding physical learning in biological tissues, although I would say we're sort of on the cusp of actually doing anything like that. So you can ask me questions. Before I go much further, I have to highlight the people who did the work. In particular, most of the work I'll talk today was done by two fantastic postdocs. Varda's now faculty, a graduate student, and a postdoc who's here, Sajjad, who has a poster. So I highly recommend you go see his poster. I'll talk a little bit about his work. A lot of the work was in collaboration with Chris Santangelo. I'll highlight the people, and then also with Angie Liu and Indrajita. So I think a lot about soft matter and biology in the intersection. And in particular, I think about the properties of large groups of cells in biological tissues. So in biology, it's become quite clear over the past 5 years or so that cells are tuning their properties in order to alter the global fluidity or rigidity of a tissue, for example, during development. And they do so in a spatio-temporally controlled way, and that is necessary. They have to get it right in order to have a proper development of the overall organism. So there's a couple of examples, one in a vertebrate, which is zebrafish, and another in fruit fly. And I'm not going to talk about any of the details for a short talk like this, but the point is that in both cases during development, you need to go from something which is roughly spherical shape to something which is very elongated. And it turns out that that's way easier if you have a fluid-like behavior of the whole tissue than if you're a solid, right? You can just think about the energy and work expended. And so the cells are clearly tuning the rigidity in order to execute this. They become fluid-like when they want to generate this large-scale flow, and then they have to solidify again because, of course, we're all solids in order to walk and be animals and do things. Okay. So the other point that I want to make is that it's not just cells. There's a lot of what are called acellularized tissues that are composed of extracellular matrix. And I think many of you, or some of you in the audience, are familiar with it. There's this feature that, of course, if I make connections in my fiber network, if I make a lot of connections, I have a lot of crosslinks, then that object is rigid. But if I start out with not too many crosslinks, so the connectivity is low, then that network is actually very floppy until I strain it a critical amount, and there's a critical strain at which it becomes rigid. So there's a floppy to rigid transition in extracellular matrices, and there's also a floppy to rigid transition in cellularized tissues. And I'm interested in those processes in general in my research group. Okay, let's see. But also biology says that all of the control systems for this process are encoded and evolve at the scale of molecules. And I want this like large scale emergent behavior of a tissue, right, which is composed of cells, which is composed of molecules. And so how do I do that control? And I'm here at this conference because I'm interested both in how does it do that, right? How does biology do that? And also kind of from a soft matter material science perspective, I'd like to make materials that can do that robustly as well. Of course, it's difficult because there are many tunable parameters, and the tuning parameters are at a much smaller scale than the emergent behavior. Okay, so that's the problem. And, the other thing I want to point out, again, I'm not going to go into any of the details in a short talk, but of course, in both fiber network systems and in cellular systems, there is a large number of what I would call candidate local learning rules. In other words, if I want to tune the behavior of a cell, there's a lot of systems, for example, that measure cell shape or that regulate tensions in cell surfaces. And then this paper, really nice paper, shows that, of course, if I have a fiber network, I can have enzymes that cleave, for example, high tension bonds, and you can think of that as a local learning rule in that network. So I'm making a kind of general point here that biology has a lot at the scale of tissues and cells to tissues, has a lot of candidate sort of local learning rules it could be using to do something. And the question is can it do something? And what is it doing? Okay. So I don't know why my pointer's not working today. Okay. The idea is that perhaps physical learning is a framework for executing and understanding this control. That's the idea I'd like to get across. And in this talk today, I'm going to have kind of a narrow focus because I want to talk about something concrete. I'm going to talk about control over tissue rigidity, fluidity, because we know that's important in these systems for like biology. And also it's useful for design. There's a lot of design principles that would be interesting if I have control over rigidity and fluidity. And then I'm going to talk about it specifically in what I'll call second order rigid systems. And I'll explain what I mean by that in just a moment. No, this is a broad audience. Okay, the rest of the talk is organized as basically what is second order rigidity and so what? Then I'm going to talk about learning fluidity and models for cellularized biological tissues. And then I'll talk about designing multifunctionality in second order rigid systems, which includes tissues, but other things too. Okay. So what am I talking about? What is second order rigidity? Okay, so rigidity has a long history of being really, really difficult to understand, right? Like, the glass physics problem, which I've also worked on, is like a big, hard problem that's still arguably not solved. Okay, and so what makes rigidity hard? Well, it's an emergent property that we take for granted, but it emerges from interactions that are very simple between objects. And so I'm just going to go through very quickly for a two-dimensional network of basically these nodes and these edges. So you can think of the edges as constraints and these nodes as having 2 degrees of freedom. They can move up, down, or left, right. So that means for a square like this, I have 8 degrees of freedom. I have 4 bars, so that's 4 constraints. In 2D, I have 3 rigid body motions, right? I have up, down, left, right, and rotation. Okay, and so that suggests that if I subtract the number of constraints from the number of degrees of freedom and ignore sort of rigid body motions, that I should have one non-trivial floppy mode and that this object should be floppy. It's not rigid. And I've drawn in the shear that realizes that particular floppiness. Okay, and of course I can fix that by just adding a bar, engineering. And so then I have 5 constraints, and then this counting predicts that there's 0 non-trivial floppy modes. Okay, but then there's an interesting thing, which is the states of self-stress, which I'll come back to later in the talk again. But the way I like to think about them is redundant constraints. So here's another set of plaquettes. And so you can see in this plaquette, I've put the 2 bars in the smart place in order to make the system rigid. But I could have decided to put both bars on one square, and then I would have this extra piece, which is still floppy. Okay, but the interesting thing is every time I introduce a redundant constraint like this, I can show to linear order that I must introduce what I'll call a state of self-stress. And that's basically, I have to have a set of tensions on these bonds that do not change the positions of the vertices. In other words, they're in the null space of the degrees of freedom, right? So, I can basically think about this as something which is a state of self-stress, and I can adjust basically a counting theory that says, okay, the number of 0 mode should be the number of degrees of freedom minus the number of constraints. But then I also have to subtract off these redundant constraints. And so I do that by counting states of self-stress. Okay, so this is known as Maxwell-Caledine constraint counting. It tells you about the jamming transition and like lots of interesting things. Okay. But there's a whole class of materials that rigidify even though they're under constrained. So the idea is that this constraint counting absolutely doesn't work for a large class of materials. And to give you an idea of what that is, I'm going to well, first I'll give you examples of like, these are the materials I'm interested in. So here's another picture of fiber networks. So you can approximate fiber networks as either a spring network or some spring networks with some bending energies. But this is a typical data set for these type of materials. So this is the shear modulus, and it's on a logarithmic scale. And so what you can see is the system has a very weak shear modulus. It's essentially floppy up until some critical shear strain at which the shear modulus jumps by orders of magnitude. Okay, and I already told you that happened, but now I'm showing you real data that says it happens. And something very similar happens in biological tissues. And again, in a short talk, I'm not going to go through all of the details, but I'll give you a flavor. So I have worked a lot on these so-called vertex models, which are not the same as vertex models from statistical mechanics, for the behavior. I didn't come up with the name, for the behavior of cells. And so the idea is if I have a layer of epithelial cells, for example, I can look at a bird's eye view of that layer of cells, and it has a cross-sectional area, and I'll call that cross-sectional area A, and a cross-sectional perimeter P. And for people who aren't biologists, you might imagine, if you got lucky, that you could write down basically a mechanical work associated with cells of a given shape, where the idea is let's say you hypothesize that cells have a homeostatic perimeter they want to control. And I'll call that sort of homeostatic perimeter P0. So it's sort of a target perimeter that they like to achieve by messing with things like cadherins and other molecules in cells. Okay. And then they also regulate either their volume or their cross-sectional area. And so they like to maintain a certain cross-sectional area. And then this is sort of the stupidest energy functional you can write down that gives you a quadratic penalty basically away from those points, from these homeostatic variables. And that's how you write it. Okay. And this is actually backed up. And I have a whole other talk where I talk about like why this makes sense and why the biology matches up with this, but I'm not giving that talk. You can ask me questions. Okay, but what you do is you non-dimensionalize it and you realize there's basically only one really important dimensionless parameter, and it's the cell shape. It's basically the ratio of the perimeter divided by the square root of the cell's area. Okay, and so what we discovered like a long time ago now is basically in these types of models, there's similarly a fluid to solid transition that occurs in these models as a function of cell shape. So if I have a large perimeter relative to my area, so it's sort of like cells kind of look elongated, then I'm in a floppy regime. And there's a critical point at which a critical shape at which the system transitions to being solid. This sort of has the same flavor as those fiber networks, right? I have a control parameter. In the fiber networks, it's the strain. In this system, it's the cell shape. Okay. And so I go to being a solid. Okay, but all of those systems are very under-constrained. They have way more degrees of freedom than constraints, an extensively large number. So how do they become rigid? Okay, and so I'm going to walk you through a simple extension of this argument. So the idea is that if I take this square and I basically sort of stretch out the geometry, so now 1 and 4 are this distance apart. You can just say I've taken this square. It has the same topology. I've stretched it out. Basically, there's another critical point when the sort of system becomes a straight line that I get a state of self-stress, and I've drawn in that state of self-stress. So I know I've broken these out just a tiny bit. You're supposed to envision that it's very nearly flat, but not quite, okay? And so then I actually have a state of self-stress, and the system becomes rigid. Basically, when this length is the sum of the other lengths, there's a critical point. Okay, and it turns out, of course, this isn't covered by constraint counting. So how do you understand this? So here's this example. Again, it has the same number as before of the degrees of freedom and constraints. And actually, now we know it has one state of self-stress. So that suggests that there should be two non-trivial zero modes in the system. And I've drawn them in here. They're the green arrows. Right? So the system is floppy with respect to movements along those green arrows. You can see immediately why sort of the argument before fails. It was a linear argument, and basically perturbations in this direction affect the bonds only to second order in the constraints. Okay, so if I only count first order stuff, I get it wrong. And so I have to consider second order perturbations to the constraints to get it right. Okay, so then the idea is, I want to understand what happens in these sort of more complex vertex models and fiber network models. And we did a ton of work. So maybe let's say there's this whole bunch of work. I don't know if any of you here are familiar with it about like rigidity theory and engineering and tensegrity and blah, blah, blah. But the idea is basically that first order rigidity is a proxy that sometimes works to tell you when a system is rigid. For Maxwell-Calodyne constraint counting, so first order rigidity is equivalent to that constraint counting that I told you about. And so it's only a subset. So if you want to know if something is rigid, you can say, well, maybe it's not first order rigid, but maybe it's still second order rigid. And maybe I can come up with tests for that. And then it actually turns out that there's some things that aren't second order rigid, but they're still rigid. Okay, so sometimes second order rigidity isn't even enough. But what we proved in this set of papers, so we wrote a set of papers, and I'm not going through any of the details because they're kind of horrible and it took a really long time, that we proved that those examples I just showed you, the fiber network examples and the vertex models, become rigid due to second order rigidity. You can prove that. Okay, so that's cool. So that means there's this whole class, and actually Chris Santangelo showed that it also is true for origami a few years back. Okay, so there's this whole class of like what I would think of as mechanical metamaterials that become rigid due to second order rigidity. And I would argue we know very little about that mechanics, but it shows up in a lot of problems we care about. Okay, so then, so maybe the conclusion to this like little vignette is that in biological tissues, second order perturbations constraints drive the rigidity. Okay, and the key point for this audience is that the tissues govern this transition via tuning of cell scale or fiber scale parameters, right? So there's this whole set of parameters that can control this transition that we really care about. Another question for this audience is, actually, this is a really cool mechanism. It's a SAT-unsat transition that's not like jamming or the glass transition. And so I also wonder if it shows up in some flavor in like chemical signaling networks or some of the other networks that people care about in this audience. I don't know the answer to that. Okay, but it's really useful because it makes you there's a phase boundary there and it makes you super sensitive, right? And you get this cool SAT-unsat transition. So if you didn't know this, maybe this is useful already. Okay, so now I'm going to ask the question, is it possible for tissues to tune this second order rigidity transition via physical learning-like mechanism? And they tune their local properties to optimize the cost function, because now we have all these variables that exist in biological systems and cells can tune them. Okay, so the inspiration for this work comes from particle packings, which are first order rigid. And so there's this work spearheaded by Varda Hegg, where we looked at particle packings. And, most of the time when you do particle packings, you have like particles that have a given radius, and then you study the mechanics of the degrees of freedom, right? Now we promote these radii degrees of freedom to be tunable variables. Okay, and so there are new degrees of freedom in the problem. And we say, oh, when we open up those new degrees of freedom and we allow optimization on those new degrees of freedom, what do we find? And so what we find is that actually tuning the correlations between the radii allows the system to find exotic ultra-stable states and basically fluidize. So we can take a state was a solid. And of course, if we open up more degrees of freedom, it's not all that surprising. If we open up these radii degrees of freedom, you can fluidize the system and the system finds these super low energy, low lying states in the glassy landscape. Okay. So it also explains the success of a new technique in glass annealing. Okay, but then the idea for tissues is what happens if we add the internal parameters as new degrees of freedom in a vertex model. Okay, and so I showed you here that there was this vertex model, it had this fluid-solid transition. And so what if we promote all of these possible internal variables that cells could potentially vary to variable degrees of freedom? Can we use those degrees of freedom to adjust global tissue mechanics? Okay, and so here I want to say first that of course, if I can tune the average values, I can do this, because I told you the system was controlled by cell shape. So if I can basically just adjust the cell shape, I can take the system through a fluid-solid transition, kind of obviously. And in fact, there's data that suggests that in fruit flies, that's exactly what fruit flies are doing. They're basically changing the average cell shape in order to, go through a fluid transition. And interestingly, in this new paper about dorsal closure, there's evidence that actually the system is working really hard to not fluidize. It wants to be an elastic system, and it actually works really hard to keep the system in an elastic regime, okay, which is kind of cool. Okay, so, that's obvious, and cells are clearly doing that. But if we want to really understand whether they can do more difficult optimization tasks, we want to say, well, what if we, just like that particle problem, so in the particle problem, of course, if I let the radii, the average radii vary, I could make one particle really big and all the particles really small, and then of course I could get a system that has very low energy. So what we're saying is, well, let's have a restriction, and this might, there might be physical reasons why it happens, that the distribution of the cell shapes has to remain the same. But can I still program this rigidity transition by varying cell shapes. And so here's just an example of two vertex model simulations that have exactly the same distribution of cell shapes, for example. So they're colored by their cell shape, but of course, they're just arranged differently. Okay. So now what we can do is we can do basically a global minimization of the energy, allowing basically these cell shapes, for example, to sort of change where they are in the packing, but not change the overall distribution. Okay. And so what we find, and the point here is that I think this is a challenging optimization task that we hope generalizes to other learning tasks. So it's the very first step. Okay, and so one of the things you can ask is, okay, if I just allowed the cell stiffnesses to vary, could I change this property? Could I change the rigidity? And the interesting thing is, no, if you change the cell stiffnesses, basically this modulus of the area or the modulus of the perimeter, you don't change the transition point. You can make it a little softer, but you don't do anything cool to this system. But if you allow the shapes to vary, you get a huge shift in the transition point, actually. And so basically the idea is you can totally fluidize one of these systems just by introducing correlations in the cell shapes instead of. So this tells you that you can do some pretty cool programming, presumably, in the space of cell shapes to modulate macroscopic emergent behaviors of tissues. And we're putting some constraints on what's possible, so we know what to do in the future for learning problems. We know that P0, these cell shapes, is really important, and also the A0s is really important. So this one doesn't work, but these two do in order to really execute learning. Okay, and so why does the transition point change? And here's these same two frames, and unsurprisingly, I had shown you two frames that have exactly the same distribution, but one of them is still a solid and the other is the fluid. And basically by learning how to rearrange, how to introduce correlations between the cell shapes, we can fluidize the tissue. So obviously there's an open question of whether cells are doing this in real life. And so I don't have the answer to that question, but it's certainly possible that they could be. Right. Another thing to say is, okay, how can I think about this? And it turns out for this set of problems, and there's a really nice paper by Max B's group at Northeastern showing that if I basically have a percolating cluster of cells that have edge tensions, then I'll be a solid and otherwise I'll be a fluid. So clearly what this rearrangement of cell shapes is doing is it's going from a network where I have a bunch of tensions along the edges to a network where those tensions are isolated to single cells, and then I don't have percolation of tension. Okay, it's pretty obvious what it's doing in hindsight. Okay, and there's an optimal, actually, standard deviation of this distribution to maximize learning. So the idea is if you say, oh, if I have like cells that have different target shapes, if they all have the same shape, I can't do this. If they have very widely varying shapes, I can't do this, but there's an optimal distribution of cell shapes. So that's pretty interesting, and it's something that you might be able to go and look for and experiment. Okay, so the conclusions for this point is biological tissues can tune their global fluidity simply by optimizing correlations between cell shapes. And that suggests that these tissues could potentially be doing learning. I don't know if they are, but I just want to throw it out there to you all that they definitely could be. So it's sort of in the tiger analogy that Eric introduced earlier, which I really like. It has the ingredients to be a tiger. We haven't shown it's a tiger, but it has the ingredients to be a tiger. Okay, and so of course, then the next steps are, is there a local learning rule that implements this minimization? And can these systems adapt, for example, to different environments using such local learning rules? Also, there's been, I told you at the beginning, a lot of observed local learning rules in the literature already. Like, here's what biological systems are doing. They're like cleaving high tension bonds. And so then, of course, you wonder, do these observed local learning rules project onto interesting global cost functions? Could we learn what the system is trying to do? Cool. Okay. So then the last point I want to make, I have like maybe 3 minutes left. Agree? Yeah, okay, perfect. So the last point I might want to make is that we're also thinking, because we think the second order rigid critical state is really interesting for lots of reasons. And so we're thinking about, can we do learning and design on the space of these second order critical states? Okay, and so the first thing I need to tell you is that for any network topology that's under constrained. So here is an example, a 3-bar linkage where I have these rotating points and I just have 3 bars. There's actually a manifold of second order critical states. And so here is like sort of a picture of that manifold for like the simplest thing I can draw. So these are the lengths of each of the bars. Okay. And so the point is that if I look at, I'm going to show you a picture now where I rotate through basically the sort of so you can see the full 3-dimensional picture for this system. Those orange points in the back there, in the back corner, they're second order rigid. The gray points are one where there's one linear zero mode, but the system is not rigid. And all the white points are also not rigid. Okay. And then the sort of the purple ones are also very interesting because those are branch points, which can also be used for things like mechanical computing. Okay. But the key thing I need to tell you is that every system, like, you know, this is the simplest one and I can show it to you, but every fiber network model, vertex model, blah, 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 that's under constrained has this beautiful manifold embedded in it. That's the second order rigid critical manifold. Okay. And when you strain an undercoordinated network, right? Like if I apply a specific strain to the system, I always drive that system to a point on the critical manifold, but it's only one point, right? There's this whole other space for a given network topology of other second order critical states. And second order critical states are really cool, right? Because I can tune an internal parameter just a tiny bit and I can go across this phase transition and I can use that to do lots of things, right? You can imagine all the things you might want to do with it. So this is an image of a random spring network that's been strained to its critical point. And I'm highlighting both sort of like the tension in cells and also features of the sort of shapes between cells. Okay. But there are, of course, other interesting states on this manifold. And okay, this is math, and I'm not actually going to go through any of it, except to say that Tyler Hain has really written down this very lovely parameterization of the critical manifold in terms of the states of self-stress. You know how I told you there was those states of self-stress? It turns out that that's a natural basis for walking around this critical manifold in any system. Okay, and so what that means is we can basically really easily walk around the critical manifold and design other functionality into a given network topology using this algorithm I'm not going to tell you about at all today, but you can come talk to me about it. Okay, so what does it mean? It means we can minimize any objective function that we can write down in terms of the states of self-stress. So for example, like you can write down like, I want the direction that's maximally stiff if I pull in this direction and minimally stiff if I, you know, in the opposite direction, for example. You can write down a criteria that you can optimize in terms of states of self-stress. Okay, so why is that interesting from a learning perspective? Oh, sorry. So here's a network. It has the same topology. So this is the same topology of the network, but I can have a random network. This one has been optimized. So it's at the critical point. So it's right at this second order critical point. And now it's been optimized to maximize the bulk stiffness. And you can see its structure looks quite different from the other one. Okay. And you can, you know, if you want to think about sort of self-assembling these types of networks, maybe you want something that has the lengths of each of the bonds is pretty similar so that you can make a bunch of objects that have similar lengths and have them attached to one another. And so if instead we take a random spring network optimized to make the springs equal in length at the critical point, actually these two are not the same, but they look pretty darn similar, which is really interesting. Okay, so I'm just giving you, there's lots here. There's lots of things you might want to optimize. I'm just saying now we can do it. So we can walk around on these critical points. And I'm really interested in thinking about learning in this space of critical points. So the conclusions there is, well, one thing I should say is all of those pictures I showed you, we did via a global optimization, because of course that's way easier than figuring out what the local learning rule is that would do it. But we expect, because these systems have interactions that are local, that we should be able to find a local learning rule. And actually we kind of know what it is for bulk stiffness. We can do it. Okay. And we can ask what other cost functions can be minimized. And we're thinking about doing learning, physical learning in the states of self-stress. Okay. So these are just a repeat of all those conclusions I told you earlier. So I'll leave you with my the people who did the work, and I'll take questions.
Thank you for the wonderful talk. I was wondering whether there's also a way in which you could apply this to predicting flows, like in a way, program flows from the parameters of the vector vertex model to design the shapes so that you get a cheaper particular flow.
So are you saying a flow in shape space or a flow of a network, a flow network?
Like flows you see in development, like in time.
Oh, I see. You're saying yes. Okay. You had a very boring cost function, Lisa. You just minimized the total energy of the system. No, you didn't say that. I'm paraphrasing. So you had a very boring cost function. You did the easiest cost function. You just made the cost function literally the physical energy of the system to show proof of principle. And so you're exactly right that one, and that's why we wanted to know which of the tuning degrees of freedom could allow us to move around in this space. And so I have no data, but that's exactly what I'm interested in is could you go and basically then say, okay, I have a task now that I want to execute like a set of flows or a set of shapes. And how would I do that in this space? Yeah, this was the first step to see which of the variables was useful for programming. Yeah. Yeah. So let me say this, which is that right at the critical point, for example, the shear modulus defined in terms of the second derivatives of the Hessian doesn't exist. But you can basically, so yes, those response functions don't exist. But you can think about actually quartic terms, and you can actually show to quartic order, there's quartic terms that actually come in and rescue the system. So you have to do different things for second order rigid systems than you would do for systems that obey a linear rule, but it's not like things go completely crazy. Yeah, actually, there's a cool paper that suggests that, yeah, in glassy systems, quartic modes can also rescue some of the behavior. Yeah.
Appreciate your talk. I was curious if there's evidence in these cellular systems for like the stiffness, like a preferred entering a preferred trait tension. I know you had a homeostatic perimeter and a homeostatic area, but that stiffness parameter in there, is there like a target stiffness that they want to enter into, or if it's for like again a preferential tension state?
Yeah, that's a really good question. And so there's a lot of different ways I could answer it. And I know I'm already over time. So let me sort of pick one and then I'd be happy to discuss others. So what I'll say is that there's actually a whole bunch, a class of lots of different vertex models. So I showed you one that sort of had a constraint on the perimeter squared, but actually you could, depending on how you feel about the problem, your emotions about it. You could also have made there sort of be an interaction term where there's a spring, for example, stiffness that sort of describes the spring interaction between sort of, you could say there's a rest length for an interface between two cells. And the reason I'm answering the question that way is it's been shown in a few really lovely papers that for certain epithelial cell types, that model works better. And there's evidence, basically, number one, that there's sort of a characteristic stiffness to that bond if you describe it in that way. And also that there's these beautiful, like, sort of local learning rules that, like, if the tension gets above a certain threshold, then the system will execute like a T1 transition, a cellular rearrangement in there. So there is certainly evidence for things like that. In other epithelial cell types where there's not this direct stiffness, figuring out what these moduli are in this model is a little bit more challenging. So I can talk to you about that.
So Lisa, I like the idea that these cells are learning through this mechanism, but could I then do something to train them, like optogenetically, like hit certain ones to soften them and see what happens? Like this to train the system to become a learning machine?
Yeah. I mean, exactly. With optogenetic tools, the question is like, what could you do? I mean, because there's one biology question, which is like, how are the systems doing that? And could you think about this as a way to organize evolvability of macroscopic functions in this system? But there's another fun point, which is like, okay, with all of the new tools that are coming, and even like, not just optogenetics external control, but like there's like sort of Rho kinase inhibitors that you can connect up to circuits with synthetic biology, right? There's a lot of things that you could think of doing to utilize this for other reasons. You could hijack it. So yes, obviously. But first, there's just so much to be done. We haven't done any of that, so yeah.